Hello again, this is Dr. Kat from Central New Mexico Community College. In this video, which is video K, on the digestive system, we're going to focus on the liver. As we discussed in the previous video, the liver receives oxygen-rich blood from the hepatic artery, with the hepatic artery being a direct branch of the celiac trunk, which in turn is a direct branch of the aorta. The abdominal aorta to be specific. But the liver also receives blood via the hepatic portal vein. That hepatic portal vein is oxygen poor, but it's very rich in nutrients because the blood in the hepatic portal vein is blood that has visited many of the digestive structures as well as the spleen, for instance. It's this blood that the liver is going to process. It's going to use some of the nutrients, it's going to remove some of the toxins, etc. While it uses the oxygen provided by the hepatic artery to go through all of these various processes. Now, when we look at the histology of the liver, we see all of these hexagonal structures. Now, of course, this is a diagram and it looks very perfect, but on a slide, it's very similar to this. The hexagons might not be perfect looking like that, but we definitely see usually 6 distinct corners with the structure in the very center of it. We refer to these hexagons as lobules. Now, one of these lobules is enlarged here. And so in the very center of our lobule, we have the central vein, which ultimately is going to lead into the interlobular veins, and they eventually become the hepatic veins. Remember, the hepatic veins come off the back of the liver and they merge with the inferior vena cava. They're oxygen poor, they're nutrient poor, they carry the blood back to the heart, the inferior vena cava, that is. At each corner of our hexagon, we have this triad, meaning we have 3 different structures, or I should say 3 different types of structures. We have one structure that is a little branch of the hepatic portal vein, so we can call it a portal venule. On the other hand, that's the blue one. On the other hand, in the red, we have a little branch of the hepatic artery, so we'll call it the portal arteriole, or I've even seen it being called the hepatic arteriole. And then we have a bile duct. Now we're, in addition to these 3, also going to find a lymphatic duct, and this hasn't been known for a long time, which is why we still refer to the corners as triads, but we do see at least one or many lymphatic ducts sitting in the vicinity of the triads. Now, what we find is that the cells of the liver is a gland, therefore the cells are all epithelial cells, are nicely arranged in these rays, almost like the rays of a sun, or in these plates, as this diagram calls them. And these liver cells we'll refer to as hepatocytes. We're going to now go to a higher magnification to better understand what is happening in between these hepatocytes and in which direction the fluids, whether it's blood or the bile from the bile ducts, flow. So here we're looking at a close-up of a few plates of our hepatocytes. This is where the central vein is located. And then at this end, we would have our triad with, you can see here, our little bile duct, our little portal venule, and our little portal arteriole, which is called the hepatic arteriole. I really prefer to use the term hepatic arteriole. Remember, in association with this triad, we often find lymphatic ducts as well. The hepatocytes have many functions, but when it comes to digestion, the digestive function of the liver is to make bile. That is the digestive function of the liver. What exactly is bile, we'll learn later, but we need bile in order to digest fats in the small intestine. So these hepatocytes are responsible for making the bile, which we see illustrated here in the green, and consequently the direction of the bile will be towards our little bile duct, which eventually is going to become the common hepatic duct. So ultimately, all the little bile ductules merge to form bigger hepatic ducts and ultimately the common hepatic duct. On the other hand, the blood flows in a total opposite direction. Remember, it is the central vein that is going to collect the blood from the liver and ultimately send it into the hepatic veins, which in their turn send it to the inferior vena cava. So the direction of the blood from the hepatic portal vein is towards the central vein, and the direction of the blood from the hepatic artery is towards the central vein. The hepatic artery has oxygen-rich blood, and consequently we have it colored in red. This blood is running in these what we call sinusoids. Remember, those are extremely leaky capillaries, very characteristic of the liver. But at the same time, we see that the blood of the hepatic portal vein, which is oxygen poor but nutrient rich, is going to also run in these sinusoids to the point that the oxygen rich and the oxygen poor blood merge with one another, and the blood in the central vein is pretty much mixed blood. So again, the direction of our blood is towards the central vein. The direction of the bile is away from the central vein towards the bile ductule, such that it can become part of the common hepatic duct. Since we have this picture in front of us, I'm going to point out one more structure or one more cell type that we'll come across, and that is the Kupfer cells. The Kupfer cells are macrophages, so these green branchy cells are macrophages that are the macrophages of the liver cell and therefore referred to as Kupfer cells. The liver has many, many functions, and we'll keep our list as brief as possible. First of all, let's start with those hepatocytes. And the hepatocytes' digestive function, as I mentioned before, is the synthesis of bile and the secretion of bile, which we need to digest the lipids that we eat. In addition, our hepatocytes play a very crucial role in removing drugs and alcohol. You might have heard that our liver forms our major detoxification center together with our kidneys. We also find that our liver can store our lipid-soluble vitamins, and I always spell them as such, K-A-D-E, which is KADE, as you pronounce it, easy to remember. It also can store vitamin B12. Now, in addition to those first few functions, the liver also metabolizes carbohydrates, lipids, amino acids, and let's talk a little bit about what that means. For one, when it comes to carbohydrate metabolism, the liver can grab the sugars that are present in our blood and convert them into glycogen. Particularly, it does that with glucose, stick all the glucose molecules together and make glycogen. Remember, glycogen is a polysaccharide. So that process is called glycogenesis, the creation of glycogen. Now, the liver can also use all kinds of different molecules to make glucose from scratch. and that we refer to as gluconeogenesis. In other words, it can grab amino acids and convert them to glucose. It can grab fatty acids, convert them to glucose, etc., called gluconeogenesis. Very tightly regulated by means of insulin and glucagon, which are two hormones produced by our pancreas. The liver also regulates our neutral fats, the fatty acids, and cholesterol levels in our blood, and also plays a role in amino acid metabolism. So what the liver can do is remove amino acids, yank off the amine group, and then we end up with urea, which is a waste product, and that we can then ultimately excrete with the help of the kidneys. But we can also see the liver using some of the amino acid present in the blood and make all new proteins, perhaps to make new hemoglobin or new clotting factors, or use those amino acids and convert them into new lipids. So now we are talking about lipogenesis, or as we talked about earlier, gluconeogenesis. So let's expand a little bit on the amino acid metabolism and talk about a process that you'll come across at times called transamination. Transamination, which often involves a protein, an enzyme called transaminase. So here we see the typical structure of an amino acid. Here we see our carboxyl group, and here we see our amine group. While here we have a keto acid. Well, the amine group can be removed, and we can then transfer it to the keto acid to now produce a whole new amino acid. The other thing is that, as is shown here, we can, from the amino acid, remove the amine group, which then can ultimately be converted to urea. The blood carries the urea to the kidneys, and then we can excrete it via our urine. So let's continue with the functions of the liver. I pointed to the macrophages in the sinusoids of the liver earlier. We refer to them as Kupffer cells. It's interesting. Macrophages often get their own unique name depending on where they're located. And aside from being your typical phagocytizing voracious cell eaters and debris eaters and antigen presenters, which I have listed here as part of the immune protection as a function of the liver, these Kupffer cells also store a variety of things. They can store iron, they can store heavy metals, as well as some lipids. Don't forget that our liver is very much involved in the synthesis of plasma proteins, and it's going to remove a variety of things from the blood in addition to the nutrients we've mentioned and drugs and alcohol and toxins. It's going to play a role in removing hormones. Hormones do not stay in our bloodstream forever. So many of these hormones that we have listed here are ultimately removed by the liver, including vitamin D3, which often is referred to as a hormone these days anyway, antibodies, and then bilirubin, which is one of the breakdown products of hemoglobin, remember, and the bilirubin is actually used to make bile. I'd like for you to read a little bit about the different forms of hepatitis in preparation for your exam. Nothing super detailed. I just want you to be aware of what hepatitis A versus B versus C is, and the things that alcoholism can do to our liver. It can destroy many of the hepatocytes to where they are replaced with scar tissue and make the liver really hard. We refer to that as cirrhosis. Consequently, the portal vein that carries blood into the liver is going to start building up a lot of pressure. We refer to that as portal hypertension, to the point that the pressure is so high that a lot of the fluid in the blood vessels, a lot of the plasma is going to start leaking out, possibly causing all kinds of swelling. Like a typical swollen belly like this is very characteristic of chronic alcoholics. We also will very often see something called caput medusae, literally meaning the head of the medusa, or similar to the head of a medusa, around the umbilical cord, meaning lots and lots of anastomosing vessels from the hepatic portal vein, again, due to that pressure that builds up. And by having these additional vessels growing, there's an attempt to reduce the pressure. We might see hemorrhoidal veins in the esophagus, and then ascites is a term that all of you should become familiar with, refers to that swollen belly that we so often see in chronic alcoholics. Let's come back to the digestive function of the liver, which is the secretion, it's the making and the secretion of bile. And what is bile? Well, I think we all know it has kind of a yellowish greenish color, but it's also important for you to realize that it's alkaline. And it's important for you to know what some of its ingredients or components are. I already mentioned it has bilirubin in it, which is one of its bile pigments. It contains cholesterol, some other lipids, even phospholipids, some electrolytes, and then there's the so-called bile salts. These bile salts are recycled. They're secreted in the bile. Once the bile has done its job in the small intestine, in the duodenum more specifically, these bile salts are actually taken back up by the blood at the level of the ileum, so that they can return into the blood, make it back to the liver, so the liver can reuse them to make new bile. We produce about 1 liter of bile a day, and notice that we can create gallstones and jaundice due to any kinds of problems with bile obstruction. By the way, your gallbladder, its main function is to just store bile. So any of the extra bile that the liver produces goes and hangs out in the gallbladder. We studied how the stomach is regulated, as well as the small intestine and the large intestine. And so we also need to learn how the release of bile is regulated, as well as the release of pancreatic juice. Both the pancreas and the release of bile by the liver and even the gallbladder are under the influence of hormones primarily. The nervous system, the parasympathetic nervous system, via the vagus nerves have some influence as well, but it's rather weak. So let's just focus on these hormones. These hormones are released when acidic and fatty chyme arrives in the duodenum. So when the duodenum receives that kind of chyme from the stomach, it starts to produce these two hormones, cholecystokinin and secretin. And you've seen me list these hormones before, but we're finally ready to discuss their functions. By the way, cholecysto always refers to the gallbladder. It's abbreviated, thank goodness, by means of CCK. When cholecystokinin ends up in the blood, eventually it's going to trigger the release of bile by the relaxation of that sphincter of Oddi, or also called the hepatopancreatic sphincter. Remember, that's the sphincter muscle that we find at the junction of the pancreatic duct and the common bile duct and the small intestine. It's also going to cause contraction of the gallbladder. Secretin, on the other hand, is going to release a bile or trigger the release of a bile as well, but this is a slightly different bile. It's going to be a bile that does not have the bile salts in it, and it's a rather watery bicarbonate rich bile. So secretin, always think of secretin or associate it with carbonate rich juice, in this case a bile. And that allows then for fatty digestion, or the digestion of fats, to begin. Remember that earlier I said the bile salts that are part of the bile synthesized by the liver are eventually reabsorbed by the ileum into the blood carries back to the liver, and the liver reuses these bile salts to make more bile. As a matter of fact, we find is that if there are more and more recycled bile salts in the blood, it triggers the liver to produce even more bile. So we have this kind of positive feedback mechanism going on. Here we see a nice image that I've borrowed from one of Pearson's books, so thanks to Pearson Publishing Company for this illustration. And we can just go through the numbers systematically. So we have, first of all, arriving from the stomach, we have arriving rather fatty chyme that's typically also acidic, because after all it's just visited the stomach. This is going to trigger the release of our two hormones, cholecystokinin and secretin. There's going to be just some weak vagal stimulation, which causes contractions of our gallbladder, so that bile is released into the cystic duct. Cholecystokinin and secretin are going to stimulate the liver to release more bile. Secretin is going to trigger the release of bile that doesn't have much bile salts in them, while cholecystokinin is going to release the kind of bile that does have bile salts in it. It also causes contraction of the gallbladder wall. Let's not forget, for the bile to make it from the liver, as a quick little review here, the bile has to enter the common hepatic duct. For the bile to leave the gallbladder, the bile has to enter the cystic duct. Where the cystic duct merges with the common hepatic duct, we form the common bile duct, which we see here, which ultimately is merged with our pancreatic duct. And at the point where these ducts enter into the small intestine, we have our hepatopancreatic sphincter muscle, or the sphincter of Oddi. This then wraps up our discussion of the liver. We didn't study the gross anatomy of the liver that much. The liver is made up of several lobes. You can take a look at that in your books. But we did study the microscopic anatomy of the liver in quite a bit of detail. And then we looked at the many functions of the liver. Remember, the main digestive function, or the only digestive function, I should say, of the liver is the making and the secreting of bile. And then we learned what controls the release of the bile. And the main control mechanism is hormonal in the form of cholecystokinin as well as secretin. The neural regulation mechanism is rather weak, but it's via parasympathetic fibers present in the vagus nerve.